Hello friends, good morning. Today I will discuss about that DNA, which is function aid, the heterocatalytic function of the translation process, which is making that protein by the help of translation. Okay, here one is that the DNA, it is one type of that genetic material which is already found in all the types of eukaryotes. Okay, and that catalyze the two types of reaction, autocatalytic and the heterocatalytic. The autocatalytic function of that DNA, the DNA replicate via semi-conservative mode of replication and the making the two daughter DNA. Okay, means that the semi-conservative mode of DNA replication that indicates that one is that parental type strand and another one is that newly synthesized strand. Here one is that the DNA replication, the function is that autocatalytic function, right? But we are talking about that heterocatalytic function. The DNA makes various types of functions. First of all, the RNA, the making the RNA via transcription. Okay, and DNA making the protein synthesis, it is involved into the protein synthesis via translation process. We are already discussing on that transcription process. Just to recall out, in this transcription we know about that transcription process which is carried out into the nucleus because the DNA, it is present in the nucleus. Okay, the transcription process, the requirement of the transcription, first of all the DNA, second one is that RNA polymerase as an enzyme, protein bridge and rho factor. Here one is that the transcription process that divides into the three steps. First of all, the initiation, elongation and the termination. And the termination sequence, it is again the transfer, again the divided into the two types, intrinsic termination and row dependent termination. Okay, these types of mechanism we are already discussing that. Please check these videos and then see that. Okay, here one is that we are talking about that in this video lecture, the translation process. This is a translation process which is carried out into that cytoplasm. Okay, transcription process, they carried out into that nucleus. Okay, and the translation process which is occur into that cytoplasm because the messenger RNA, it is moves to that cytoplasm and making that protein synthesis. Okay, here one is that the translation process requirement. The first one is that mature messenger RNA. Okay, the first requirement is this mature messenger RNA that encodes that various types of genetic code which is related for that protein synthesis. Second one is that as a carrier molecule of the transfer RNA. Third one is that activated amino acid. We know about that protein, it is making by that various types of amino acid which is binds with each other via peptide bond. It is called that CONH bond, right? And then making that long polypeptide chain, it is called that protein, right? So that activated amino acid, it is most requirement for that protein synthesis. And fourth one is that the ribosome, it is called that protein factory. And in this translation process we are discussing about that prokaryotic translation. So that 70s ribosomes, it is part in this process, and we know that the 70s ribosomes made up by that two subunits. First one is that 50s subunit and second one is that 30s subunit. Okay, here one is that the translation process, it is divided into that three steps: initiation, elongation and the termination. Initiation having that three factors: initiation factor 1, 2 and 3, and then elongation having that three factors: elongation factor Tu, elongation factor Ts and elongation factor G. And then termination is the ending the process of the protein synthesis that contains that three factors: releasing factor 1, releasing factor 2 and ribosomal releasing factor. Okay, this is all about that translation process. We are focusing on that translation process in the detail. See, here one is that the diagram of that translation process. Here one is that translation. We are talking about the first step of that initiation. Okay, here one is that the initiation process, which is having that ribosome. Okay, the first of all, the 30S ribosome containing the three sites. First one is that exit site, P means that peptide bond formation site, and A means that amino acid binding site. Okay, the mature messenger RNA, it binds with ribosome via Sine-Dargano site. Remember this site name, Sine-Dargano sequence. ओके, मीन्स यहाँ पर क्या होगा, तो मैच्योर मैसेंजर आरएनए रिबोसोम पर बाइंड होगा साइन-दरगानो सीक्वेंस से, और देन आफ्टर दैट स्टार्टिंग द इनिशिएशन एंड आल्सो कॉल्ड एज अ प्रोटीन सिंथेसिस. We know about the formyl methionine. It is a starting character and also called as a starting factor for that protein synthesis. It contains that AUG, means that methionine codon. Okay, we know about that AUG. Here one is that the initiation factor that requires the three factors, initiation factor 1, 2 and 3. यहाँ पर क्या होगा, तो 3 साइट्स हैं, अलग अलग साइट्स पर. तो पेप्टाइड जो ट्रांसफ़र आरएनए है जो लेकर आया है एक्टिवेटेड अमाइनो एसिड, उसके 3 प्राइम OH ग्रुप पर है. यहाँ पर होगा, आपको पता है ना कि ट्रांसफ़र आरएनए का स्ट्रक्चर कैसा होता है. ओके, तो ट्रांसफ़र आरएनए के स्ट्रक्चर पर 3 प्राइम OH पर एक्टिवेटेड अमाइनो एसिड क्या हुआ है, बाइंड हुआ है. ओके, एंड देन क्या होता है, तो इनिशिएशन प्रोसेस शुरू होती है. यहाँ पर क्या होगा, तो फर्स्ट ऑफ ऑल इनिशिएशन फैक्टर शुरू होने से हमें पता है कि एमिनो एसिड बाइंड कहाँ पर होगा, ए साइट पर. लेकिन यहाँ पर क्या होता है, तो इनिशिएशन फैक्टर 3 दे क्लोज द एग्जिट साइट एंड देन इनिशिएशन फैक्टर 1 and 2 क्लोज द ए साइट, मीन्स दैट अमाइनो एसिड बाइंडिंग साइट. सो only P it is a open in for that protein बाइंडिंग. ओके, प्रोटीन से बाइंड होता है एंड देन आफ्टर दैट 70S कॉम्प्लेक्स जब फॉर्म होगा तब क्या होगा, तो इनिशिएशन फैक्टर 3, 1 and 2 रिलीज आउट हो जाएंगे, और देन ओनली फॉर्माइल मिथ्योनिन दैट्स बाइंड्स विथ P साइट. ओके, दिस इज ऑल अबाउट दैट इनिशिएशन प्रोसेस. ओके, then after we will talking about the elongation process. The elongation process, it is that अगेन यहाँ पर भी 3 फैक्टर्स हैं. एलोंगेशन फैक्टर TU यहाँ पर फर्स्ट आएगा, फिर वो लेके आएगा एक्टिवेटेड ट्रांसफ़र आरएनए को, और वो यूज़ करेगा ATP को. ATP यूटिलाइज एंड देन मेक ADP प्लस PI, एंड ऑल्सो दैट कंटेन्स दैट फॉस्फेटिडाइल ट्रांसफरेज एंजाइम. तो फॉस्फेटिडाइल ट्रांसफरेज एंजाइम के तौर पर क्या यहाँ पर होता है कि एक्टिवेटेड ट्रांसफ़र आरएनए फॉर्माइल मिथ्योनिन के साथ क्या बनाता है, पेप्टाइड फॉर्मेशन करता है यहाँ पर. तो पेप्टाइड फॉर्मेशन होने से यहाँ पर पेप्टाइड फॉर्मेशन हो गया. ओके, एंड देन पेप्टाइड फॉर्मेशन होने के बाद आता है सेकंड फैक्टर, एलोंगेशन फैक्टर TS. दे रिमूव द एलोंगेशन फैक्टर TU. ओके, एंड देन अगेन मूव दैट एलोंगेशन फैक्टर G आता है, और एलोंगेशन फैक्टर G क्या करता है, तो 5 प्राइम टू 3 प्राइम मैसेंजर आरएनए को मूव कराता है. ओके, एंड देन क्या होता है, तो जो अनचार्ज ट्रांसफ़र आरएनए है, ये अनचार्ज ट्रांसफ़र आरएनए एग्जिट साइट पर जाता है, और जो पेप्टाइड बॉन्ड कंटेनिंग ट्रांसफ़र आरएनए है वो P साइट पर आता है. ओके, बाय द हेल्प ऑफ GTP. एंड देन this type of process that moves again and again, and then making the polypeptide chain. Here one is that you can see that polypeptide chain formation. Okay, ऐसे ही polypeptide chain यहाँ पर again the activated transfer RNA that binds with A site, and then again the peptide bond formation occur, and then these types of process repeatedly they start. Okay, here one is that then after that polypeptide chain formation occur, that termination process, it is very important for that protein synthesis stopping. Okay, here one is that the termination process, again the 3 factors involving releasing factor 1, 2 and ribosomal releasing factor. Okay, releasing factor 1 that contains that three types of characteristics. First of all, that it is like that transfer RNA, so that it binds with पी साइड सॉरी अमाइनो एसिड बाइंडिंग साइड पर क्या करता है, रिलीजिंग फैक्टर बाइंड होता है. ओके, एंड सेकंड कैरेक्टरिस्टिक इज दैट it binds with it is also contain the endonuclease enzyme, so that the cleavage that polypeptide chain into that restriction sites. And third one is that it contains that termination codon. The termination codons क्या करते हैं, तो termination करवाते हैं. ओके, then termination codon, after the termination releasing factor 1 it's released when binding with releasing factor 2. जहाँ पर releasing factor 2 आता है तब क्या होता है, तो releasing factor 1 रिमूव हो जाता है. एंड देन एलोंगेशन फैक्टर G आता है, वो क्या करवाता है कि 5 प्राइम टू 3 प्राइम मैसेंजर आरएनए का मूविंग करवाता है. और देन जो अनचार्ज ट्रांसफर आरएनए है वो एग्जिट साइट पर आता है. ओके, here one is that ribosomal releasing factor, and then after the ribosomal releasing factor यहाँ पर जुड़ गया है ट्रांसफर आरएनए से, अमाइनो एसिड बाइंडिंग साइट से यहाँ पर जुड़ गया है. अनचार्ज अमाइनो एसिड यहाँ से एग्जिट साइट्स पर क्या होगा, रिमूव हो जाएगा. देन आफ्टर